stabilized for the most part. Okay, well, welcome everyone. It's wonderful. This is our fourth session, and to have it on World Cancer Day is a pleasure. I think this is going to be a really nice presentation by all of the speakers. My name's Megan Mahoney, and I'm the Director of Scientific Affairs and Training Programs at BioPyramid. And I'm very pleased to welcome you to this session. So for those of you who are new, we launched the Summit Speaker Series as an opportunity to bring together our community of different types of stakeholders in the absence of a face-to-face summer for cancer immunotherapy. And so, as I mentioned, this is our fourth session, and we plan to continue this until we have our next Cancer for Immunotherapy event in 2020 November. And so with that being said, I welcome all of our speakers today. Today's talk title is titled Therapeutic Inducers of Natural Killer Cell Killing. ThINKK is all you need to know, right? It's a novel approach to prevent cancer relapse after hematopoietic stem cell transplantation. Now, before we begin, I'd just like to highlight our next two sessions. The next session is by Carolina Elkow, and she'll be talking about her engineered biovesicle platform. And that's taking place February 22nd. And of course, as always, we'll be sending out this material on our social media. And then the subsequent talk to that will be taking place on March 10th, and that's by Dr. Schonfeld and Jennifer Beasy, who will be giving a little bit more of a biomanufacturing 101. So what does it take to manufacture a vaccine? And specifically, they'll be talking about it from a biomanufacturing perspective. So just moving on to a few housekeeping notes before we begin. This session will be posted on our website afterwards. And if you'd like more information about today's session, including the speaker bios, please do go to our website. The information will be linked in the chat box momentarily. In addition, if you have any questions for the speakers, you can ask them via the Q&A box at the bottom of your screen. And you'll be able to type them in throughout the session. And then we'll ask them at the end at certain points. And what's nice about this function is that you can upvote specific questions of interest. And finally, if you're having any sort of technical difficulties, please use the chat function. So there's a Q&A function for questions and the chat function for technical issues. So, as I mentioned, we're having three speakers today. The first is by Marie-France Langlais, and she's the mother of Lucas, who is a child who was diagnosed with acute myeloid leukemia, and I'll let Marie-France describe that. And of course, she'll be presenting a patient perspective, and in particular, a caregiver perspective. Following Marie-France will be Nicolas Poirier, who's a master's student with Michel Duval and presenting, a plain language summary of the ThINKK data. And then at 2 o'clock, we'll have Michel Duval present on his ThINKK data. And Michel is a pediatrician and hematologist-oncologist, a clinical professor with the Department of Pediatrics at the Université de Montréal, and the head of the CHU Saint-Justine Department of Hemato-oncology. Those French words were tripping me up. But with that, I'd like to stop sharing and pass it over to you, Marie.
Good afternoon, everybody. Welcome to my home, actually. I hope everybody can hear me well. Like Megan told you, I am Marie-France Langlais. I'm the mother of a former patient of Dr. Michel Duval. Today, I will talk to you on how I ended up getting involved in the ThINKK project with Dr. Duval. So I'll start with a little bit of history first about my family. I have a wonderful husband, Don, and two amazing, beautiful children, Delphine and Luca. The story that how I got here started back in 2004. Almost 17 years ago today, as a matter of fact, on Friday, February the 13th, my son, Luca, age 9 at that time, got diagnosed with acute myeloid leukemia at the Montreal St. Justine Hospital, for that matter. Back then, we were told his chances of surviving 5 years to such a diagnosis was around 25%. Needless to say, the shock, the pain, and the anxiety were unbearable. That day, the monster entered in my family. Treatment started with 3 rounds of aggressive chemotherapy. The protocol that should have applied was calling for 5 rounds of chemotherapy, followed by bone marrow transplant only if chemotherapy would have failed. Lucas suffered a complication to the heart due to one of the chemo involved in the induction, the doxorubicin actually, in that induction first treatment. So consequently, his treatment protocol was revised and reduced to 3 rounds only. We left the hospital in June 2004. He was in remission, even though he didn't have the full protocol. We were hopeful. Maybe the monster was gone. Fortunately, 9 months after his diagnosis, in October, that matter, Luca didn't feel good again. A bone marrow puncture proved he was relapsing. This time, though, we could not avoid it. We had to proceed with the bone marrow transplant. There was no other option available. Ultimately, after some more chemo and some more associated complications, pneumonia, pancreatitis, and all sorts of other consequences, the team was able to transplant him with cord blood in January 2005. Luca left the hospital in March, feeling reasonably good again and in remission. Again, we were hopeful. Again, maybe the monster left my family. Already we had gone through down a very rough path of dealing with the cancer monster for more than a year. A very, very, very long year in the life of a child and his family. Luca then entered the process of being monitored on a monthly basis at the walk-in clinic of St. Justin. Let's face it, if we were to go there every month to be tested, he was at high risk of relapsing. Unfortunately, PCR came back positive a few times, even though chimerism proved to be close to 100% donor at each visit. Michel then recommended, I will always remember his words, a somewhat experimental treatment, administering interferon alpha a few times a week over a period of almost 2 years. I remember him saying, "It is not proven yet, but I'm hopeful. And it's promising." So naively or desperately, I guess, I did not want to hear the part somewhat experimental, not proven as being effective yet. I only wanted to hear promising. I wanted to hear, let's boost the bone marrow transplant to be stronger in the fight against traces leukemia. Oh yeah, I'll buy that. I don't think either I wanted to admit that this was probably the ultimate chance of surviving the cancer monster. Well, the interferon worked. The new marrow too. Luca ended up being followed very closely by Michel until the age of 20. Today, he's 26 years old. He's healthy, he's happy. He's free of cancer. No apparent sequel, enjoying his life as an actor, as a normal human being to the fullest. The monster is dead. When Luca graduated from St. Justin, cured and healthy, that last visit with Michel at the clinic, I turned to Michel to hug him and thank him. I candidly told him, anything you need from me, anytime, I will always be there. I'm just a phone call away. So Luca left St. Justin that day, but I never did. Michel and his team called me a few weeks later, in February 2013 actually, to get me involved in a continuous improvement committee for the bone marrow transplant program. From there, I got involved in numerous other partnering with patients and parents projects and initiatives across St. Justin and elsewhere in Montreal. Ultimately, the CHU St. Justin even offered me a position as senior advisor leading the Bureau du Partenariat Patient Famille Soignant de St. Justin in January 2018. In the fight against against pediatric cancer. Families are constantly exposed to the change of plans to the treatment, to important temporary or permanent side effects or sequelae, to hope and despair, to other families losing their child. To this day, some other families are waiting for a miracle to beat the monster. They are looking for proven solutions that will make their journey somewhat safer and hopeful than the tightrope walking I felt we had to do for years, hand in hand with the medical team. The ThINKK project that Michelle is leading is extremely promising for such families. That is why I and other patients or parents, partners like me are on board to support this project and collaborate with Michelle and the other researchers. Moreover, we are excited to collaborate in this project with the Marvel project, the Marvel team from the hospital in Ottawa. We are in, so other families like mine will suffer less, and also to know in our hearts that we contribute to their safer journey. Only then, maybe will I be able to graduate from St. Justin too. Maybe you want to show the pictures, Megan? So I would like to thank Michel and his team who got me on board on this project. I would like to thank you all for your support. I would like to thank you all for your dedication to this cause. We are all in this together against the monster. Thank you.
Again, we don't hear you. Again, we don't hear you.
Yeah, we didn't hear properly, but I think
I apologize.
No,
I was on mute. I apologize. Yeah, if you wouldn't mind going ahead, Nicola, and sharing your slides.
Yeah, sure. That's what I figured you had done. So just bear with me for a second while I share my screen. There we go. Well, so first I'd like to thank you, Marie-France, for this great presentation you just did, putting us back in the context and really reminding us, well, why we do the work that we do. It was truly moving. I love that. Thank you. So, hi, my name is Nicholas, and I'm part of Michel Duval research team. And today I'll be making a brief introduction to the work that we are doing in his lab. So basically what we've been doing is working on an immunotherapy against acute lymphoblastic leukemia by adaptive transfer of activated in vitro differentiated plasmacytoid dendritic cells. So quite a mouthful, but we're going to break that down in a few minutes. So it should be quite simple. So first and foremost, leukemia is a type of cancer that affects blood cells. So we call that an hematological cancer. And basically it initiates in the bone marrow, and it affects a process that our body uses to produce new and renew the blood cells called hematopoiesis. And when we say acute lymphoblastic leukemia, so ALL, it means that it basically affects the lymphoid lineage of that hematopoiesis process that is responsible for the production of T lymphocyte, B lymphocyte, and NK cells. It is often further It is often further separated into subtypes of ALL. So, like, for example, pre-B ALL, which is the most common type we hear about because it represents 80% of all the ALL. And in the case of a leukemia, it's usually more than 20% of those cells progenitors, so like the one circled there, T cell progenitor, that are considered to be lymphoblastic. They're lymphoblasts, so basically cancer cells. We've diagnosed 6,150 new cases just in 2020 of acute lymphoblastic leukemia. So it's a quite common cancer, and more than 80% of patients are between 1 and 18 years old. So it's predominantly a childhood cancer. So basically, as Marie-France kind of put us in the context, but the first line of defense we have against acute lymphoblastic leukemia, ALL, is chemotherapy, as it is for many cancers out there. And it's separated in 3 different stages. So induction, consolidation, and maintenance. The first step of induction is the most intense. That aims to really break down the leukemia to the lowest level that we're able to get it. So inducing what we call the remission. So basically a state where the leukemia is at a very, very low level. And then we can transmission, when we have reached remission, we can transition to consolidation that can last between 2 and 4 months. Where we lower a bit of the intensity of chemotherapy, but we try to keep lowering that number of residual leukemia cells, so we can further reduce the intensity of chemotherapy and get into maintenance for the 2 or 4 years later, so we can keep that remission going all along and prevent that leukemia from resurfacing. Sadly, as we all know, this process is not 100% efficient. And sometimes relapse can occur. So the leukemia can resurface even though we keep on administering chemotherapy. And at that moment, our next line of defense, our next clinical option is hematopoietic stem cell transplantation, or as Marie-France called it, bone marrow transplant can also be referred to as. So allogenic hematopoietic stem cell transplantation, that we'll call allo-HSCT, is basically we sample LT stem cell from a donor or from cord blood that we inject into the patient to allow its system to reconstitute from that LT base. And in the hope that that reconstituted system will be able to clear the residual leukemia on its own. Following it, so the main effect that allows that allows to clear that leukemia is something we call graft versus leukemia effect, so GVL. And the main effector of that effect are NK cells and cytotoxic lymphocytes that we'll come back to later. Sadly, following allo-HSCT, as high as 40% of patients can relapse. And the allo-HSCT is the last therapeutic alternative for high-risk patient. So as it was the case for Marie-France, the only thing you can do after allo-HSCT is turn to more experimental treatments such as interferon-based immunotherapy, putting an highlight that we need. Well, we think we need a more efficient allo-HSCT or an alternative treatment for those high-risk patients that relapse from the graft. So just to summarize NK cells, some of you might know them already, or might have heard the name, natural killer cells. They're basically the sole bearer, the bearer of innate immunity. So our body's ability to defend against pathogen and infection. NK cells can be finely tuned between using a wide array of receptors that allow them to either recognize cells that belongs to our body and inhibit themselves and not kill those cells, or recognize, by example, tumor cells that have a lower expression of self-recognition molecules, or infected cells, stress cell, and induce cell death, so induce cytotoxicity. And it's that process that allows NK cells to get rid of those leukemia cells that are present in the body. So if we go back to the graft context, when we administer the graft, the immune system of the patient is at its lowest. So all the cells need to be reconstituted from the stem cell that we administer during the graft. And the first one to do so are the NK cell, who can, as early as 2 months after following the graft, as shown right here on the graph, can be back to a normal level in comparison to the T lymphocytes that won't reconstitute before 3 months and can take up to 12 months before being back to a normal level. So when we look at that in terms of the effector of the GVL effect that I mentioned before, that is the key effect we want to aim for, the NK cell in the first few months are pretty much the sole bearer of this this heavy task that is clearing out leukemia. So this is why we think increasing their activation or activating those cells could really help in the fight against leukemia. It has been shown that ALL tend to be resistant to that cytotoxic, to that NK cell induced cytotoxicity. And this phenomenon has also been linked with higher relapse from the graft. So this is why we've been working on that kind of side of the problem. So what we thought about doing is increasing NK cell cytotoxicity against ALL, so we could overcome that resistance that sometimes occur in ALL and reinforce that GVLs to reduce post-transplant relapses. And the way we chose to do that is by harnessing the power of a cell called plasmacytoid dendritic cells, so PDCs. And PDCs are naturally produced by our system. They represent 0.1% of PBMCs. Well, PBMCs are basically just the entirety of our immune cells, and they are innate activators of NK cells. So they have a very unique ability to produce incredibly high amounts of interferon alpha, which was the molecule the immunotherapy for Marie-France kid was based on. And by using that molecule, it can directly activate NK cells and then increase GVL effect. So what we believe would be possible is by using PDCs in the context of an HSCT, hematopoietic stem cell transplantation, we could increase the GVL effect of the NK cells and reduce post-transplant relapse. So what we've done in our lab is basically we've designed a technique to produce in vitro a cell that we entitled Therapeutic Inducers of Natural Killer Cells Killing, so ThINKK, which are a close cousin to the PDC that we know in our peripheral blood. They share all the phenotypes of pretty much all the phenotypes of the PDC, and they keep that ability that they have to produce very high amount of interferon alpha, and so activate NK cell cytotoxicity against leukemia. And it's that mechanism that is core to the immunotherapy that we are proposing to help the fight against acute lymphoblastic leukemia. So basically, just to summarize the approach that we're taking, we'd be administering, following the graft, weekly injection of ThINKK, which is our activator, and could increase that NK cell cytotoxicity to increase the GVL effect and reduce post-transplant relapse. So basically, this is really the core of what we're aiming to do. So we're constantly working on various sides of this problem to ensure that everything's set up for it to work perfectly. But yeah, this is the core of our research and our project. And finally, before I leave the spotlight to Michelle, if just for people who are more visual, or if people didn't catch a word that I just said, all you need to remember is that what we've done is we've designed a cell that we call ThINKK that, following activation, can produce high amounts of interferon alpha, and if transplanted in the patient, could activate NK cells to increase their ability to lyse leukemia lymphoblasts and get rid of the disease. So that's the core of what we're doing. And I'll just leave the spotlight to Michelle to go more in depth with how we're doing that, or how we're achieving our goals. So thank you.
And perhaps, Michelle, as you're setting up, I don't see any questions in the Q&A, but of course we do have a few minutes before 2 if there are questions for Nicolas or for Marie-France. So I do invite questions, whether they're from a patient perspective or any questions that may help you understand the presentation that Michelle will give. So I'll just give it a minute or 2 to see if any come in. But other than that, looks like we're set up. So I suppose no reason not to get started, and perhaps save some more times for questions at the end. And of course, we'll open it up to Q&A at the end as well. So with that, why don't you take us off, Michelle? And I will say you're on mute. I'm not sure if you know that, but just a reminder.
Yes, I just was trying to unmute myself, and because I can't have both the access to the microphone and the pointer, so no, I have both. Yeah, thank you very much for the opportunity of presenting, and hello to everybody. It's really bizarre to have to speak to people you don't see and you don't know who they are, and you're not even able to greet before the presentation. So don't hesitate to ask questions. I will try to be relatively short to give time to the questions at the end. So as Nicolas told you, the approach we are putting forward is called Therapeutic Inducers of a Natural Killer Cell Killing, ThINKK. And I think by the end of the presentation, you should be able to understand why we have called them that way. To begin with, doesn't work. To begin with, I want to thank everybody, all the people who have worked on that project over the years, because it's the last part of a long project who began in the almost 20 years ago. Almost people in the lab, people who helped me in the clinical research unit, and people also, all the colleagues in the group de recherche in cord blood transplantation that were set up in Saint-Justine that lead us to many things, including that one. And also, I want to thank all the institution and corporation and everybody who helped us to send us money to be able to do those experiments. And particularly, I want to thank one person, Paolo Cordero, who is a technician in the lab. He was in the lab even before I came in, so he is very important for me and for the research we are doing. First, childhood cancer. Everybody knows that the progress has been made in childhood cancer, and I just want you to look at the blue line here. The blue line is the mortality rate of childhood cancer. When you look in the 70s, it was very high, or at that time, almost 80% of the children died of their cancer, and it decreased slowly until the mid-90s. Since the mid-90s, that's almost 30 years ago, the rate of the decreasing of the mortality was still there, but it was much lower than it was before. And if you continue the curve, you will see that today it's almost the same improvement since the last 15 years. This nice curve here was almost only attributed to chemotherapy. At that time, chemotherapy was had given all it has to give, and the small progress we see since 30 years are almost progress due to immunotherapy. And in immunotherapy, there is many ways to do immunotherapy, but the one we are doing the most usually is hematopoietic transplantation. It's called also hematopoietic stem cell transplantation or bone marrow transplantation, although sometimes the graft is not bone marrow, it can be cord blood, as in Lucas' case. And why is it an immunotherapy? It's because what we are doing, we get rid of all the hematopoietic system of the recipient. Hematopoietic system means the blood cells and the cells who produce that blood cells. The cells who produces the blood cells are the marrow cells. So we get rid of all the marrow cells, and we get rid of all the blood cells of the recipient of the graft. And then we engraft what we call a stem cell, hematopoietic stem cell. It's not an embryonic stem cell, it's an hematopoietic stem cell. But for us hematologists, we call that simply stem cell. And that stem cells give rise to all, by differentiation, to all here, the cells that you can find in the blood, including the red cells, also the white cells, macrophage, polynuclears, and also three particular cells who are constituting the immune system. The B cells does not interest us here. It's the cells who are producing antibodies. It's not very useful against cancer, but it's part of the immune system. And the two cells we are interested in against cancer or leukemia in transplantation are NK cells, natural killer cells, and T cells. That if both of them can induce what we call a graft versus leukemia effect, or graft versus tumor effect, or graft versus cancer effect, both from NK cells and T cells. But T cells also can induce graft versus host disease. Graft versus host disease means that the T cells who are coming from the stem cells we transplant are coming from the donor, and they attack the recipient. They are attacking all the normal cells of the recipient, and that can be very dangerous for the recipient, and even can lead to death from the transplantation. So from the beginning of my career as an immunologist and a pediatric oncologist and transplanter, I was very interested with NK cells, because NK cells were able to induce graft versus leukemia or graft versus cancer effect without inducing graft versus host disease. As I told you, there is still progress to be done in the treatment of severe leukemia, and that's a study we published with colleagues all across North America a few years ago, showing that in acute myeloid leukemia and acute lymphoid leukemia, acute leukemias comes in two types: lymphoid, lymphoblastic, and myeloid. In acute myeloid leukemia and acute lymphoid leukemia, the survival curve was very poor in people with high-risk features before transplantation. For those who don't never seen a survival curve, it's important that I show you, because you will be some others in the presentation. You see here the time in years, and here the proportion of patients who are surviving, meaning that in that situation, when your risk score is at 3, at 1 year post-transplant, there is only 15% of the patients who are surviving. And when we look at 2 years post-transplant, there is less than 5% of patients surviving, meaning that overall there is progress to be done in those two situations. Immunotherapy can be also by other means, and in another childhood cancer, which is called neuroblastoma. It's not a brain tumor, it's another tumor, but it's a tumor that kills many children a year in Canada and over the world. You can see with such a survival curve, that was a study we participated in, showing that with standard therapy, at 2 years post-transplantation, there is 46% of patients still surviving, whereas at 2 years post-transplantation, where they receive immunotherapy, and I will tell you which immunotherapy it is, there is 2/3 of patients still surviving, showing that in these situations, immunotherapy is given also after transplantation. In this situation, when we give 2 cytokines, cytokines are proteins who generally activate some cells of the immune system. So we will give 2 cytokines, IL-2 and GM-CSF, and a monoclonal antibody after transplantation for neuroblastoma. It improves the survival, showing that indeed post-transplant immunotherapy can improve the survival of children with cancer. So the NK cells was interesting for many reasons. Another reason is that it does not rely on recognition of the cells on the HLA matching, for those who know the HLA. So they can apply on any HLA background, meaning that it works for almost for everybody, not depending on the ethnic background you are from. So the NK cells, there is on the surface of the NK cells, as Nicola told you, there is many receptors. I will just ask you to remain 2 of them, the trail one and the kill one. If we look at the trail one, when that receptors encounter a ligand on the tumor cells, the NK cells is activated. The same way with all those ligands, they can be activated just by encountering their ligand on the tumor cells. And those also. NKG2A and KIR are special because when they encounter their ligand, they are not activated, but desactivated. And usually that relation is important because most of the cell of our body have the ligand for KIRs, meaning that our NK cells, when they found the ligand for KIR, are desactivated and they do not attack our normal cells. So just remind of both two of them, TRAIL, which is an activating receptor, and KIR, who are a desactivating receptor. TRAIL is a little bit more complicated than that because it's both activate the NK cells and tell the tumor cells to die. We will see that. So it's an important receptor on NK cell. 20 years ago, another proof of the influence and the possibility to harness NK cells after transplantation has been done by an Italian team who take donors that are mismatched for KIR with the recipient. If they are mismatched for KIR, the KIR on the donor NK cells do not have any inhibitory signal. That is, the desactivation is deactivated. When the desactivation is deactivated, the cell is activated and it can kill the tumor cells, not only the tumor cells, myeloid leukemia, but also cells that play a role in graft-versus-host disease and in rejection of the transplant. So the hypothesis was, if we do it properly, when we have this mismatch between the donor and the recipient, which is not possible for many patients, but it's in some situations can be realized, then we will have less relapses, less leukemia post-transplant, less graft-versus-host disease, and less relapse. And indeed, the first results from that team show that in acute myeloid leukemia, when there was a mismatch between the donor and the recipient in the right way, there was no rejection, no graft-versus-host disease, and for AML, acute myeloid leukemia, no relapse. However, for acute lymphoblastic leukemia, there was relapse. And from that time on, everybody thinks and says everywhere that acute lymphoblastic leukemias are resistant to the NK cell killing, particularly after hematopoietic stem cell transplantation. We had also personal experience, as Marie-France told us soon, and perhaps she will remember that story. So this is a story of a young boy who was transplanted at 9 years old. He was transplanted day 0 of the transplant. Before the transplant, leukemia was found in the blood and in the marrow. I should have run marrow here. And after the transplantation, almost all children receive cyclosporine to prevent the graft-versus-host disease by calming the T cells. So he received cyclosporin, and one day, around day 60 post-transplant, we found slight amounts of leukemia in his blood, not in his marrow. And then we did it again, and indeed it was positive twice. So we decreased rapidly the cyclosporin in the hope to let the immune system to fight the leukemia. And it worked initially, because the leukemia, we searched it by PCR, that is able to find very little amounts of leukemia, became negative again in the marrow and in the blood. And then we found it positive again. And at that time, I was trying to convince my colleagues across North America to do a clinical trial with interferon alpha in those situations. And in the situation of that young boy, which you have recognized as Lucas, the son of Marie-France, It was special because he received cord blood, so we were not able to go back to the donor to ask for cells for immunotherapy. And at that time, the success rate of second transplantation was very low. So the only classical option in his situation was to just wait until leukemia recurs frankly, and then give him chemotherapy again, and then go to transplant. So I told Marie-France, I don't know if I use the term experimental, but I told Marie-France that we were working on setting up a study, but it was not open yet, but I had hope in it. So we decided together with Marie-France, Don, Lucas Fazer, and Lucas to try that interferon because it was relatively safe. It was given subcutaneously 3 times a week or twice a week, I don't remember at that time, for 2 years. And sometimes it can give a few symptoms like flu symptoms, but not more than that. And indeed, when we gave interferon alpha, it came back again to negativity, and we gave interferon alpha for 2 years. I know we are more than 15 years, and because it's going well, and you see his picture during Marie-France's talk. So that was showing that indeed it could work. I never succeeded to convince my colleagues across North America to do that trial. It has been done a few years after, 10 years after, by a Chinese group who showed that indeed interferon alpha was able, in some cases, particular cases, to induce remissions when the leukemia was recurring at low level, like in Lucas, but it was not a constant effect, and it was not. It was reproducible, but it was not enough to make a big difference in the relapse rate. And indeed, when we use also after cord blood transplantation, when we looked at the difference between patients who are KIR mismatched, so with NK cells activated, and KIR matched, so NK cells deactivated, we can see that in acute myeloid leukemia, indeed, there is a big difference, because at 3 years post-transplant, you can see in when the NK cells are activated, almost 70% of the patients are still alive, whereas at 3 years, when NK cells are not deactivated, rather, it's almost 30% of the patients are still alive. It's true in acute myeloid leukemia. It's not so true in acute lymphoid leukemia. So, but there is a difference, and this difference is seen also in other studies too, showing that maybe there can be something to do with NK cells against acute lymphoblastic leukemia. And in the lab, as well as with other researchers from Saint Justine, we indeed have had at that time some argument making us think that acute lymphoblastic leukemia, contrary of what people think, was able to be killed by NK cells if these NK cells are properly activated, and KIR mismatch or interferon alpha was probably not sufficient. So at that time, if once we said that interferon alpha was not sufficient, we look at which cells are producing the interferon alpha. And indeed, most of the interferon alpha produced in the body is produced by the very rare cells who are called plasmacytoid dendritic cells. We call them PDC because plasmacytoid dendritic doesn't mean anything. Plasmacytoid means round. These cells are indeed round, and dendritic means exactly the contrary. What are those cells doing is when they sense the presence of a virus, any virus in the body, they activate. They become round like this. They produce a lot of cytokines like interferon alpha. And in this state, they activate NK cells. That most of the PDCs do that. They act as soon as they sense a virus, they activate. They produce cytokines, they activate NK cells. Some of PDCs, on the contrary, became dendritic cells, and they activate not NK cells, but rather T cells. And as I told you to begin with, we don't like T cell activation after transplantation, because T cell activation can lead to death by graft-versus-host disease. So we were very interested, not with all plasmacytoid dendritic cells, but rather with the real plasmacytoid cells, those who are around and activate the NK cells. So RON, NK cells activator, dendritic, meaning dendritic, I think dendros means something like finger or hair, I don't remember in Greek, but those hairy cells are T cells activators. And recently, a few years ago, with the techniques as are called single cell sequencing, all the dendritic cells of the body have been studied by the Brian study by Villani as first author. And you see here all the dendritic cells. So dendritic cells are usually here to trigger the immune system. So there is what we call the DC1, DC2, DC3, DC4. Perhaps I don't tell them in the good order, but those four are DC1, 2, 3, 4. A new population described by Villani as DC5, and another population which calls DC6. DC6 is very interesting for us because it's what we can call the real PDCs. So if we talk about dendritic cells, the one we like are those one, DC6, because they are really plasmacytoid dendritic cells, whereas these one are real plasmacytoid cells activating NK cells, but also T cells, whereas all the other are T cell activators. And another study that had been published a few months after showed that there is possibility when you take the plasmacytoid, the PDCs, and you activate them, you see that they can segregate in 3 subpopulations. A population is called P1 with PD-L1 positive, CD80 positive, no matter. But one which P1, these P1 are the real round cells producing cytokines, whereas the P3 are those dendritic cells activating T cells. And the P2 are intermediate, where they are also all together producing cytokines and activating T cells. So when you take the PDCs before activation, you activate them with something that mimics a viral nucleic acid. Those cells goes all here, or here, or here, P1, P2, P3, and the one we like here and we want to produce is rather the P1 cell. So P1 in that experiment, and DC6 in that experiment. And what is the summary of what we wanted to do? To have PDCs, activate them with what we call TLR ligands. We can answer questions about that, but mainly they are mimicking the presence of a virus. Those PDCs produce a large array of large amounts of cytokines, including and mainly interferon alpha, but also many other cytokines. Those cytokines activate properly the NK cells, and our hypothesis was if they are activated properly, they will kill not only acute myeloid cells, but also acute lymphoblastic cells. And indeed, Martin Leledier, who was a master student in the lab, showed that it was true. He took 3 different lineage of lymphoblastic cells. And here, for those who are not familiar with that, it's what we call cytotoxic assays, meaning if you take 5 NK cells against 1 leukemia, 1 against 1, 2.5 against 1, and 5 against 1, and you see how many leukemia cells are killed by those NK cells. And you see that when you do not stimulate the NK cells, nothing happens. When you stimulate the NK cells, as we did for Lucas in this situation medically, you have a little bit of increase of the killing of the acute lymphoblastic leukemia. But when you stimulate the NK cells with not only interferon alpha, but all the cytokines produced by the PDCs, you have a very large killing of the lymphoblastic cells, and that was true for the 3 lineage that was studied by Martin. And I just want to show you something. Trail, I talked to you about trail, and we discovered that that way, that novel way, not described by others at that time, to activate NK cells was very particular because the NK cells increased their expression of trail as they do not in any other mean of to activate them. So it was very specific to the activation by PDCs, this high expression of trail. That expression of those molecules were less specific. It's seen in every NK cell activation. The trail expression, overexpression, is very specific of that activation by PDCs. And Martin Cordeau, who was at the same time, we had Martin and Martine in the lab as master students. He was working on leukemia, acute leukemia, and she was working on neuroblastoma. Shows the same, that the expression of trail was very specific of the activation of NK cell by PDCs. And Martin, using another model, but we'll talk about later, but in neuroblastoma, showed that indeed when we use interferon alpha, we were able to kill the targets, but when we use the PDCs, we were able to kill the target more efficiently, although it was depending on the production of interferon alpha by the PDCs. So the killing of our targets depends on interferon alpha production. It needs interferon alpha production, but if we give interferon alpha, it's not enough. It's not just, it doesn't resume the whole effect. And what Martin showed also, that if we take a line that do not express a receptor for trail, we do not see any killing. And when we take lines that express receptor for trail, we see a killing. And when we block the interaction of a trail with these receptors, in most of the case, we see the disparation of the killing of the leukemias, meaning that trail is very important in that killing. So we wonder why, and we knew that from studies for other teams, that when trail is expressed by here you are inside the tumor cells, and here you are inside the NK cell. So when trail is expressed at low level on the NK cells, the NK cell says to the tumor cells to die by the way of what we call the mitochondrial pathway of the apoptosis. And when trail is expressed at high level on the NK cells, it bypasses that mitochondrial for the direct pathway of apoptosis, leading to the tumor cell death. And indeed, when we take out one gene of that cascade out, we see that the killing was almost completely disappeared. And so we looked at our bank at Saint-Justin on almost 200 patients having ALL, of the expressions, but RNA, the expression of those cascade. And we saw that indeed the expression of caspase-9 here is decreased in the patients, whereas the expression of all the other pathway here is normal. The idea is probably that in acute lymphoblastic leukemia escapes normal NK cells when they only express trail at low level, but they cannot escape NK cell killing when trail is expressed at high levels. Most cell lines are sensitive. This is a work that's done by Emily, who is a doctorate, a PhD student now in the lab. Most of the cells are sensitive, but some cell lines, as you've seen also 697, are not sensitive. So she's working on the way to find what we call biomarkers, that is what on the acute leukemia cells can tell us before making the experiments, can tell us if the acute leukemia cells is resistant or sensitive to our approach. It's important because if we're going to give that to patients, it's better to select the patient that will really benefit from the approach and not give it to patients that for which the leukemia will be resistant to the approach. So she's working on that definition of biomarker of resistance and sensitivity to our approach. So back to transplantation. Émilie Charrier, she was also a PhD student in the lab. She studied the NK cell reconstitution after transplantation. Here you see months after transplantation, 1 month, 2 months, 3 months, up to 1 year after transplantation. In gray here, it's the normal level of NK cells. And she compared bone marrow transplantation without triangles with cord blood transplantation, the round. There was a lot of variability, but most of the patients had normal NK cells number at 1 or even 2 months post-transplant. I don't show you those results, but she published also that those NK cells were not only normal in numbers, but also normal in functions and even increase in a subtype who is known as having anti-tumor effects. So NK cells are active, present and active from 1 month post-transplant. If you look at the transplantation, NK cells during the 2 first months are active and present and active, whereas T cells are much longer, take much longer to go back to normal, more than 1 year post-transplant. When we transplant the patients, we give them chemotherapy before the transplantation. The leukemia burden goes very low, and relapse occurs when this leukemia burden increases. So we are very interested in that therapeutic window because, as I show you in Lucas' case, all those immunotherapeutic approaches works much better when the leukemia burden is very low. So during the 2 first months, it's very interesting because we have high levels of NK cells, anti-tumor that can be used to be anti-leukemic. We have low levels of ALL disease, which is more amenable to immunotherapy effect. And we have low level of T cells, which is good because T cells, when activated, can lead to graft-versus-Host Disease. So we are very interested in that windows from, I would say, 6 weeks to 12 weeks post-transplant. So Emily looked at the reconstitution of PDCs, those triggers who normally activate NK cell after transplantation. And I don't show you the result for bone marrow, but in bone marrow transplantation, there was no reconstitution up to 1 year post-transplant. Whereas in cord blood transplantation, there was no normal number of PDCs, but the normal numbers of PDCs were absolutely hypofunctional, not functional. They do not produce interferon alpha until 1 year post-transplant. So if we go back to that figures, we cannot use normal reconstituting PDCs during that therapeutic windows because it would have been fine to have altogether NK cells, no T cells, low acute leukemia disease, and PDCs, because if we, in this case, it would suffice to activate the PDCs recovering from the graft, but it's not the case. We have no PDCs during that period. So the only way to activate NK cells during that period of time, that therapeutic window, is to activate them with PDCs we would take from people and give to the patient. And indeed, what we did, we took such PDCs and we used what we call a preclinical model. It's a mice in which the blood, all the blood cells, the marrow cells, etc., the immune system is completely humanized. It's a human immune and hematopoietic system in a mouse. So it's a closer system you can get from the patient when talking about transplantation. So what Liliane did, she was also a master's student in the lab. She put together with the help of Eli Haddad a model of that transplantation. So newborn mice irradiated, like it's just like the conditioning regimen of the transplant, given was CD34, it's stem cells basically. And then after once the reconstitution was begun, she gave a little amount of acute lymphoblastic leukemia cells. And then or nothing, or those activated PDCs. All the cells, the amount, the number of cells we choose are always chosen to be the same as what we see in children. Of course, mice are much not so as big as children, but we use a conversion system or based on the surface of the body. And what she saw that when here you have the control group, so the group who received everything but not the activated PDCs, and that control group, when the mice goes red, meaning that the leukemia, which is fluorescent, is about to kill the mouse. So in the control group, all the mice were turning red. In the IL-2 group, which is another cytokine who activates NK cells, also they are turning red. In the PDCs, where the PDCs were not activated, they are also turning red, whereas in the group that received activated PDCs, all the mice survived. This is a survival curve, a little bit different. So you see that when you give in IL-2 or nothing, all the mice die from leukemia, whereas when you give activated PDCs, all the mice survive. So this is a closer we can do from the patients, and the next step after that is clinical trial. We'll talk about it. The problem with PDCs, that I told you in beginning, they are very, very rare. So we will not be able to take PDCs from somebody and give it to somebody else because the numbers are very too low to be extracted from somebody. So Sabine in the lab, she's an associate in the lab, research associate in the lab, or as a research associate. She devised a new way to try to differentiate PDC analogs, saying analogs because it's not real PDC. She take stem cells from cord blood, and two rounds of expansion with a receipt I will not give you to and to anybody else. She is able to grow a big amount of PDCs. The PDCs or PDCs analog are able to produce high amounts of interferon alpha when they are activated. So they are PDCs, they look like PDCs and they sing like PDCs. They're probably not exactly PDCs, but PDCs analog is a good term to begin with. And we look at them at with the single cell approach, like I show you. Just look at that one. We show that the bulk of the product of those cells are very similar. They are segregating in the same subgroup. And there are some subpopulation. One here, which is probably a less mature population because it's cycling, as you say. It's cycling, and it's probably the population from where all the cells mature. This one is a more mature population here. So altogether, all those cells here, the red, the green, and the blue ones are like PDCs analog. This one looks a little bit like what we call DC5. We don't like it very much. And those DC5 indeed express a rather specific marker, which is called AXL. We'll talk about it. And those small amount of cells here are cells that have nothing to do neither with these dendritic cells. PDCs are just contaminants. So the cells are very homogeneous, and most of them are from looks like DC6 cells, the ones we like. And a little amount of them express the marker AXL, which is correlated in normal cells with the property to activate T cells, and we don't like that very much. And indeed, when we look at that P1, P2, P3, you remember we like the P1, we don't like the others. Indeed, those cells, those PDC analogs, all goes into the P1 window, which is NK activating. None of them goes in the P3, which is T cell activating. A few of them goes into the P2, which is both of it. And when we look at production in interferon alpha, we see that almost all cells produce some degree of interferon alpha. So we have cells that are really like the plasmacytoid compartment of plasmacytoid dendritic cells, the round ones who produce cytokines, activate NK cells, but do not activate T cells. Nicolas was involved in just looking at that. So we will just look at that part here. When you do not stimulate T cells, the subgroup is called CD4, it's true for the other subgroup CD8. When you do not stimulate NK cells, nothing happens, no worry. When you stimulate NK cells with very strong stimulus, then when you stimulate the T cells with a very strong stimulus, CD3, CD28, then you see the T cells are marching, or we call that marching to the left, meaning that they are losing the fluorescence we put it in them before. So they are marching to the left. So when the cells are marching that way, it means that they are proliferating. So when we give a very strong stimulus of proliferation, they proliferate. We are happy. When we give the natural stimulus of proliferation, which are dendritic cells, but T cell who activate T cells, indeed they also march to the left. When we give our product, they don't march to the left. They don't proliferate. And when we add our product to the natural activator, it's a little bit less. It's not significant, but it's a little bit less. So at least they do not increase the proliferation of hemodisease. And Valérie Paquin, she was also a master student in the lab, and she worked on the same approach. Just to look at that, it's a model of Graft-versus-Host Disease, and we looked at two populations, a population of mice who did not receive PDCs, and a population of received PDCs, and there is no statistical difference between the two groups, meaning that in vivo too, the PDCs analog we had do not exacerbate increased graft-versus-host disease. So we decide to give a name to those PDC analogs because they are not exactly PDCs. They are characterized by the property of activating mainly NK cells. So we call them therapeutic inducers of natural killer cell killing, or ThINKK. We are very proud of that name. It's like a flag you can fight under. Nicolas in the lab also looked at the AXL population, and he showed that when you take the AXL positive population, so ones we don't like very much, you have a relative amount of T cell proliferation, not as much as the natural stimulus, but a little bit of it. And if you give the AXL, you take the AXL negative population, then you don't have that stimulus. So we are trying to improve also our production of cells with several molecules. I won't tell you the real name, but with several molecules that produce in the final product much less AXL positive cells that we do not like because they activate, they might activate T cells. What we did also is to verify that when we put them with T cells from somebody else or NK cells from somebody else, they are not destroyed by another immune system, which is important because if we do that, we can use those ThINKK from another person to give to a patient. So another person different from the recipient and the donor of the transplant. And also we test different conditions of cryopreservation. That is, we froze the cell and see if they are able in number and in function to be still active if we freeze them and then thaw them again, which is important because it's much more easy to use if you have the cells frozen than if you have to prepare them and give them without freezing. We even use our mice model, preclinical mice model, to determine which is the best schedules, and the best schedule is to give 30 millions of cells by meter square. A meter square to a child of 10 years is or around 1 meter square body surface area. So it's relatively low number of cells, 10 times less that is used usually in immune adaptive systems, because one of these cells is able to activate 10 NK cells. And also, what did Nicolas? He looked at the effect of immunosuppressive regimen, because as I told you, after transplantation, in Lucas' case, for example, he received immunosuppressants to calm the T cells to prevent graft-versus-Host Disease. And he showed that in the case of tacrolimus, mycophenolate, and sirolimus, there was no difference. So our system worked even if there is those molecules in the patient's body, but it do not work when the patient receives cyclosporine. So when we will go to the clinical trial, we will exclude patients who receive cyclosporine or ask that they receive another immunosuppressive agent. And that's the future clinical trial we want to go to. The same figure as shown by Nicolas. A patient with remaining acute lymphoblastic leukemia, he undergoes acute stem cell transplantation. A little bit of leukemia remains. At that time, we have prepared the ThINKK. That are frozen, thawed a few days after the transplantation, and given to the patients to exactly as we did in the mice to prevent leukemia relapse. I will go a little bit faster on the last slide, just to see that we have seen exactly the same effect for another killing tumors of children, which is called neuroblastoma, with an increase of a bright increase of neuroblastoma cell killing by our approach. Also, in that case, trail was important, but another ligand on NK cells was also important, which is called DNM1. And when we add the immunotherapy, I was talking about anti-JD2, we see that our approach is indeed it increases the effect of the regular immunotherapy that already exists. And we show that even our approach was successful in vitro, even if we take the NK cells of the patients again his own tumor. That was done by Asila Belounis, who was a PhD student in the lab. And Marina Ayoub, who was also, she made a stage in the lab, showed that, and we are working on that also, showed that indeed it works very well on acute myeloid leukemia, which is not surprising because everybody knows that acute myeloid leukemia is indeed sensitive to NK cell killing, and which shows acute lymphoblastic because it was resistant. So she proved that indeed it works with AML. So this our project, our future of the projects lies in that slide. We want to go to the clinical trial, and with the help of, in particular, biocanrics, we hope to be there. We hope to be there in 2 years from now. I'm finished. If you have questions.
Wonderful. Thank you for that, Michel. So I'd invite questions to continue to come in either by Q&A, and I see a couple in chat. So I'll start with one question, and that is, trail can be upregulated on neutrophils using interferon alpha. Out of curiosity, did you by chance check neutrophil cells activation?
No. No, neutrophils doesn't go into our system. It has never been shown that neutrophils can have an impact on the relapse of leukemia after transplantation. So we never had a look on neutrophils because of the clinical data that shows that there is no correlation between neutrophil reconstitution and relapse, for example.
Okay, great. And sorry, I'm just looking through here for questions. There are a few comments and questions. Okay, great. So another question, are there two cytokine therapies administered concurrently, or excuse me, are the two cytokine therapies administered concurrently or sequentially? I'm asking from the perspective of immune cell activation and how it may be advantageous to activate immune lineages in a specific order.
Oh, yes, that's a good question. The answer is in the neuroblastoma trial I showed you, indeed, there was a sequential activation, GM-CSF, a rather complicated sequential activation, GM-CSF, then IL-2, then the antibody, very complicated. And another team has tried to just take the IL-2 away because it was toxic. IL-2 gives like a super flu. They took the IL-2 away and it worked the same way. So yes, there is, usually people think that it's important to put that in the right order, but it may not be true. No, I
apologize. I mute myself in between because there's a little bit of
just to finish
noise.
the answer, indeed, when we use adoptive cell therapy, is one of the goals is just to have the cells giving the cytokines in the good order, rather than to try to guess in the lab which is the good order and give the cytokines. In our case, we give the plasmacytoid dendritic cells, and we hope that those cells who are done for that, they evolved to do that, they will give the proper cytokine at the proper time in the good order.
Okay, great. So the next question from the same person is along the same lines. Are there any thoughts or ideas or work on pre-treating the bone marrow donor cells prior to transplant, such as treating with agents that which may ablate specific immune lineages, for example, T cells capable of clonal expansion and persistence?
Yes, of course. That has been done for, I would say, 30 years now, to what we call T cell depletion. So taking the bone marrow, taking out the T cells and re-injecting the bone marrow. In that situation, you have much less graft-versus-host disease, or almost no graft-versus-host disease, but you have much more relapse of the leukemia. And at the end, the results in terms of survival is the same. So the children do not experience, or the adults do not experience graft-versus-host disease, but they relapse in a much higher number. And almost until recently, this approach has been abandoned for by almost everybody. It has a sort of rebirth now, because we use we can use, if we properly take the T cells out, we can use the parents to give as a donor for the transplant. But in that situation particularly, we still have a big problem of relapse. So we need to increase, if we take the T cells out, we have to activate the NK cells if we do not want to have relapse.
Okay, great. And there are many questions coming in, which is wonderful. So I thought perhaps to your point about engagement, maybe if anyone has any questions they'd prefer to ask orally, then I might suggest you raise your hand and then I'll be able to unmute you. So that's another option that we can trial out for today's session. With that being said, I will continue through our chat here. The next question is, how long do these PDCs persist in your mouse model, and do they remain functional throughout that time?
The answer is not long, but we don't know exactly. Not long, probably not more than a few days, because we show that we have to inject them twice a week if we can to have the maximum effect. So it probably means that they are lasting less than 3 or 4 days. But we are not able to tracking them in the mice. We have tried to do that, but they are in so little numbers that we have not been able to track them in the mice. So we don't know where they are going, and we don't know how long they are persisting in the mice. But in our model, it's probably around 2 to 4 days.
And what is the
which
planned
is
route of,
which is the normal
sorry.
lifespan of an activated PDC. An activated PDC, generally those who activate NK cells last 2 days, and those who are activating T cells not more than 4 or 5 days.
Wonderful. Okay, and the next question is, what's your planned route of administration for the PDC, intravenous or intradermal?
Intravenous. Intravenous, because the leukemia is the cells. If you inject the cells intradermal, they will have great difficulties to go into the body. And in the case of leukemia, the leukemic cells are in the marrow or in the blood, so they are very easy to target when you inject them intravenously.
Great. And let me just check the chat box. I've been looking at the chat or the Q&A. Okay, great. So there is another question in Q&A. Have you checked the effects of applying ThINKK-derived exosomes in ALL instead of ThINKK?
Of what exosomes derived from?
ThINKK-derived exosomes.
We have tried to pursue that way of research, but it has been very disappointing. It's a very good question, but we have not been able to really reproduce the results. And I think it was Asila Balounis, so one of the students, the PhD student in the lab. She lost time, months, if not years, trying to do something properly, and so we abandoned that way. But it's a very good question. Yet it may be as efficient as exosomes or small vesicles in general, that the whole cell, but too complicated.
Okay, great. Perhaps you touched on this, but would it be optimal in terms of scheduling to start with GD2 followed by GM-CSF, and then dendritic cell activation to scavenge, etc. Perhaps something that may spare CD4 T cell lineage that may be useful to keep?
I'm not sure I understood the question.
Yeah,
I think to use cytokines.
If I can interrupt just a second.
Yeah.
If I can interrupt just a second, I think it was because I think it's Tarun Sanda which asked the question, the two questions you answered at the beginning, but she was sending them as you were answering. So I think it was follow-up. So when you did the first two comments, so would it be optimal to start with GD2 followed by GM-CSF belongs to your answer concerning the administered concurrently or sequentially. But then the second comment talking about perhaps something that may separate CD4 lineage is about pre-treating the bone marrow. So those are separate comments.
Okay.
So that's your question. I guess the question, would it be optimal to start with GD2 followed by GM-CSF? That would be one as a whole.
Nice to have the students to explain me what the problem is.
I wasn't answering, so I had time to follow through.
Thank you for inviting him.
and yeah, to respond and saying perfect. So thank you for that too. Did you want to add anything to your answer, Michelle, or you think you covered it in your previous responses?
No. I think I covered it in the previous responses. Anyway, the idea is to give cells, because those cells will be able to give the right cytokine at the right moment on the right place. And when we give GM-CSF or IL-2 to patients with neuroblastoma, they are very, very, very sick, very, very sick. Because we give the cytokines all over the body, that activates everything everywhere. Whereas where we send the PDCs, we hope, and it probably like this, it works. If they find the NK cells, they produce the cytokines just where the cytokine is needed, in the right amount which is needed, in the proper order. And it's make a whole difference in the term of safety.
Perfect, thank you. So we're at 5 minutes. I don't see any more questions coming in, which I think that's good. I think you answered quite a lot of questions, so thank you for that. At this point, I again invite questions for any of our panel or our speakers rather. So I'll give a minute or two for that. Maybe just as a final remark from my end, just a reminder to please fill out the survey at the end of the session. So when you close out of the screen, another one should auto pop up, and it's quite a brief survey, and it just helps inform our sessions, and we really appreciate the feedback. So again, I don't see any questions. So maybe if I could have maybe final comments, maybe one final comment or thought for the audience from each of you. Quite open-ended, so I leave it to you to interpret that question.
I've talked too much. Marie-France, Nicolas, help.
And I don't want to put you on the spot. So if you think you covered everything, which I think you sufficiently did, I would certainly say that. Perhaps no, Marie-France, no. from your end, from the patient and caregiver perspective.
Well, I think I've said it also in my speech at the beginning, but I want to reiterate, you know, how for all these families that are awaiting a miracle, all these families, you saw the numbers of families behind these numbers, behind all those graphics that Michelle showed. There is a family, there are families that lose their child. We have to remember that. So in the name of all these families that are going forward and trying to survive what happened to them, you know, we have to be thankful that some people continue, you know, to find a cure for the monster, to kill that monster. So that's all I have to say. I'm always grateful to be here still, to be with my child still, but I am very, very fortunate. So let other families be this fortunate. That's what I have to say.
I think that's a wonderful note to end on. So thank you for that poignant message. Again, I'd really like to thank you all for presenting. I think it was such a wonderful synthesis of the information, and really wonderful to have you here. And of course, I'd like to thank all of our participants for attending. It's great to have you despite it being a virtual and very bizarre setting. So thanks everyone. And with that, we'll end here and have a great day. Take care, guys.
Yeah.
Thank you.
Bye now.
Thank you.
Bon.
Yeah.